Today's topic is mechanism of DNA replication in prokaryotes. Cells without nuclear envelope are prokaryotes. DNA replication is a process by which a double-stranded DNA molecule is copied to produce new identical DNA molecule. DNA replication in prokaryotes occurs in cytoplasm. It occurs in cytoplasm, whereas in case of DNA replication in eukaryotes, it occurs in nucleus. DNA replication in prokaryotes is always bidirectional and semi-conservative. The three basic steps involved in DNA replication is initiation, elongation, and last one is termination. First, we start describing the number one, that is initiation. Replication starts with the binding of initiation protein to the double-stranded DNA molecule. It leads to the formation of replication bubble. The replication bubble is the region where purine and pyrimidine are attached to each other. Initiation protein along with ATP helps in the melting of hydrogen bond and create the replication bubble. The function of replication bubble is to attract an enzyme, helicase. Helicase recognize the replication bubble and gets bind to it. The function of helicase is to make the replication bubble bigger by unwinding the DNA to remove and another enzyme called gyrase. Gyrase goes ahead of helicase and its activity resolves the single-stranded binding protein which binds to the template strand and makes sure that the two strands do not reunite together. The next one is the step, that is elongation. The elongation of the new strand starts with the RNA primer, the DNA polymerase III, which binds with primer and start to synthesize a new strand. The synthesis of new strand is always from 5' to 3' direction. No trouble occur in the strand going from this direction in the replication fork and produce the leading strand. The strand that goes to the opposite direction of the replication fork, it faces some trouble. Due to the facing of the trouble, its synthesis is in fragments, which is called as Okazaki fragment. And the DNA polymerase first remove the RNA primer and replace the gap with a nucleotide, and then another enzyme called the DNA ligase. The DNA ligase glue the Okazaki fragments and leads to the formation of lagging strand. Now the mechanism should stop, and this is the termination phase, where the termination protein called TUS protein binds with the terminating sequence and blocks the movement of helicase. It inhibits the activity of helicase and no synthesis of the new strand occurs in the absence of helicase. In this way, we have finished the topic and it's very simple to memorize. This is the chart. This is the chart. This is the chart, and thanks for watching. If you like this video, please like, subscribe and comment.